I also wanted to thank the organizers for inviting me to this great meeting. So we will move a little bit into the gut now. And firstly, I will talk about how the microbiota diversity is generated and the role of IgAs, immunoglobulin A, in this process. And then I will move into how the bacteria are kept away from the systemic circulation. So the establishment of the microbiota occurs in the first days and years of life. And in particular, you are born more or less sterile, but then later on you acquire the microbiota primarily through first the delivery, through the mothers, and through lactation from the mother. So this is an important issue because through the milk, the mother is also delivering a lot of IgAs, immunoglobulin of the A type, which are the secretory immunoglobulin. And what is the role of these immunoglobulins in controlling the differentiation of the microbiota? So we know that the microbiota interacts a lot with the host. In particular, the microbiota is found in the outer mucous layer, which is present primarily in the terminal ileum and in the colon. And in the outer mucous layer, the microbiota is colonizing. And you can see here, this is in red, is the microbiota. You can see in green is the mucus, primarily in MAC2, which is the major mucin in the gut. And you can see that there are two regions of the mucus. An outer mucous layer, that is where the microbiota is residing. And there is an inner mucous layer, which is almost sterile, you can see here. And the reason being that there is a lot of antimicrobial proteins that are released and also of immunoglobulins of the A type. So as I said in the beginning, the seminar structure is divided into two parts. So first, how is the microbiota diversity generated? And the second part, what controls the systemic dissemination of bacteria. So regarding the first part, as I said, immunoglobulins of the A type, they are very important. I mean, one of the major recognized function of these immunoglobulins is that of immune exclusion. So the bacteria are bound by the IgAs, and they are kept away from the gut. But another important function of the IgAs is to allow the bacteria to bind to the mucous layer that I told you is colonized by the bacteria. So this function is important primarily for those bacteria that are not capable of binding the mucus by themselves. And so in order not to be washed out from the gut during the peristaltic movements, they have to be attached to the mucus. And one way through which they are attached is via the IgAs. So another function of IgAs, which is important, is the capacity of IgAs to mediate a controlled entrance of the bacteria. So the IgAs neutralize the bacteria. This allows them to be kept away, but also it allows through receptors that are still unknown, those bacteria to be internalized. And since they are covered with IgAs, so they are completely opsonized, these bacteria are not capable anymore of virulence, but can still be internalized. And what is the function of this mechanism was not known. So the focus of the first part of the presentation is regarding the function of IgAs to internalize the bacteria and also to allow the bacteria to bind to the mucous layer. So if you look at different animals, so mice of different genetic background, we know that black 6 animals, so they have very much lower amount of IgAs both in the serum and in the feces. By contrast, if you look at bulb C animals, they have much higher amount of IgAs both in the serum and in the feces. And this correlates with the diversity of the microbiota. So the lower the amount of IgAs, the lower the diversity as compared to the higher amount of IgAs, higher diversity. But this diversity is not only of those specific IgAs, but also for polyreactive IgAs. So you have two types of IgAs, the innate IgAs that are polyreactive and the specific IgAs which are specific for bugs. The polyreactive IgAs, they can recognize common components of bacteria. And if, for instance, if you take animals that have never seen Salmonella, if you look at IgAs that are specific for Salmonella, they're reactive for Salmonella, you can see that they are already present in bulb C animals and very few in the black 6 animals. So animals that have higher IgAs, they also have higher polyreactive IgAs. And they can bind also bacteria that they have never seen before. So this was our initial hypothesis, that you have natural IgAs that are polyreactive. These natural IgAs are released, and they can bind to bacteria independent on the bacteria just because they recognize common features of these bacteria. And if they allow the internalization of these bacteria, even if these bacteria are not capable by themselves to be internalized, like for instance most of the microbiota, which is not invasive, then once it's inside the host, these IgA-bound bacteria can be recognized by the dendritic cells, which activate B cells. And now these B cells can be specific for these bacteria because they can recognize antigens that are specific for those bacteria. And now you have a second wave of IgAs that are not polyreactive, but they are specific for those bacteria that have been internalized. And these IgAs can allow now these bacteria to bind to the mucus. So the more natural IgAs you have, the more antigen-specific IgAs you will have, and the more diversified these will be, and the more of bacteria that bind to the mucus you will have. So you will have increased the diversity of the bacteria just because you have more IgAs that will be binding to this bacteria and will allow these bacteria to colonize, to attach to the mucus. So is this hypothesis true? What we have been doing is the following. So we have taken mice that are germ-free, so they don't have microbiota, and we have colonized these mice either with a fecal transplant coming from bulb C animals, so to reconstitute the bulb C animals, or vice versa, the the black animals of C57 black 6, together with the feces coming from their own conventional mice. Or vice versa, we have done bulb C animals transplanted with black 6 feces, and vice versa, black 6 animals transplanted with bulb C feces. At different time points from fecal transplantation, we have collected the feces, and we have analyzed for IgAs and for microbiota, just to see whether we could reconstitute the original mice, and what happens when you have bulb C animals that normally has more IgAs colonized with the C57 black 6 microbiota, and vice versa. So the first thing we found was that normally we think that these animals that are germ-free, they have very few IgAs. And this is true if you look at black 6 animals. But it's not completely true if you look at bulb C animals. So even if they don't have the microbiota, you already have some IgAs that are produced in the bulb C animals. By contrast, the black 6 animals have very few IgAs. And when you colonize these animals, so at week 2 already, you can see that there is a higher amount of IgAs produced in these animals. And this, in the beginning, is independent on the microbiota. But then if you put the bulb C feces into the bulb C, you have a higher amount of IgAs as compared to the black 6. So you have a genetical predisposition to produce IgAs, which is independent on the microbiota. And then different microbiotas can induce different types of amounts of IgAs. While if you look at the black 6 animals now, the scale is much reduced because of the bulb C, but you also see some increase in IgA production at later time points during colonization. So then what happens if you look at the microbiota? So are these IgAs determining the diversity of the microbiota or not? So as I said, we took the stools and we did DNA extraction from the microbiota, and we analyzed by 16S rDNA hypervariable region amplification and deep sequencing the microbiota by Illumina. And this was quite striking because what we could see is, as I said in the beginning, the bulb C have a higher diversity of the microbiota as compared to the black 6. This is the alpha diversity. And when we look at different time points, let's look at the week 8, which is the last week. The bulb C with the bulb C microbiota has higher diversity, and as compared to the bulb C with the black 6 microbiota. This is because you start with a lower the lower diversity of the microbiota. So even if you put it in a higher amount of IgAs, still you cannot increase the diversity. But what is very interesting is that if you put the bulb C microbiota, which is much more diverse, into the black 6 mice that have few amounts of IgAs as compared to the bulb C, then the diversity is reduced as compared to the bulb C. So the amount of IgAs, and you can see it here because there is a direct correlation between the amount of IgAs and the diversity of the microbiota. So the amount of IgAs and the diversity of IgAs is controlling also the diversity of the microbiota. So our hypothesis is confirmed. We also had a lot of other data, but this is published if you want to go into details. That you have a genetical determination of how the amount of IgAs and the diversity of IgAs that is produced in natural IgAs. These natural IgAs can bind to the microbiota in the initial phases of life and allows this internalization of the microbiota. And once inside the host, then you have production of antigen-specific IgAs, which are increasing the amount of IgAs that can bind to the microbiota, and since these are specific, also increases the diversity of the microbiota, which is also important for the stabilization of the microbiota. And this allows the microbiota, which is not capable of binding the mucus by itself, to bind to the mucus and to colonize. So the higher the amount of IgAs, the more diverse is the microbiota, and the more diverse is the microbiota is the best, let's say, healthy status of an organism. So going into the second part of my talk is, as I said in the beginning, once you have several lines of defense towards for the host versus the microbiota. And the first one is the mucus layer. So the mucus layers keeps the bacteria away from the epithelium. But it can happen that there are some bacteria that are mucus degrading, and they can cross the mucus, and some bacteria also are capable of crossing the epithelium and getting into the lamina propria. However, if you look at the difference on how antigens are taken up by the host, we know that if you have soluble antigens can get in our host and they can disseminate through the lymphatics into the mesenteric lymph nodes, where most of the tolerance towards those antigens is established. But it also you can find the same soluble antigens in the liver, where CD8 tolerance is established. So soluble antigens can gain access both to the lymphatics and to the portal circulation and get into the liver. And then from the liver, they can also get towards the systemic circulation. So soluble antigens are internalized by the host, and they get systemically circulating. And this is important for establishment of oral tolerance. So if the same antigens are entering the host through a different route, they are tolerized. By contrast, if you look at the microbiota, especially in the mouse system, the microbiota is entering the host and it gets stuck into the mesenteric lymph nodes. Andrew McPherson has shown very nicely this. And the mesenteric lymph nodes are like a firewall for the microbiota, and so the microbiota does not enter into the systemic circulation. However, this for us it was quite striking, because if you look at how the gut is organized, so you have these are the intestinal villi, you have the epithelium, which is just lining as a single layer towards the intestinal lumen. But if you look at the vessels, you can see that the blood vessels, so those that will get into the portal circulation, are much closer to the epithelium as compared to the lymphatics. So the lymphatics are much deeper into the lamina propria. So how come that bacteria can get easily into the mesenteric lymph nodes, so they reach the lymphatics very easily and get into the mesenteric lymph nodes, but they are excluded from the blood vessels, which are just sitting here? So we hypothesized the following, that similarly to the brain, in which you have a blood-brain barrier, also in the gut you have a vascular barrier that we call the gut vascular barrier that is selective in controlling what are the antigens that get into the systemic circulation. So if you look at the brain, in the brain you have endothelial cells that are sealed by the presence of adherence junctions and tight junctions. They don't allow the passage of molecules between the cells. And this structure is generated and it's organized into a vascular unit, which is called the neurovascular unit, which is made by the endothelial cells, the pericytes, which are surrounding the endothelial cells, and the astrocytes and neurons that have end feet that are controlling the activity of these cells. So also in the gut, we have endothelial cells, blood endothelial cells that are sitting just below the epithelial cells. And we know that here we have the equivalent of the astrocytes, which are called enteric glial cells. So we wondered whether also in the gut we had the gut vascular unit, which was made by endothelial cells and maybe the enteric glial cells, which are the equivalent of the astrocytes, and what were the endothelial characteristics. So do they have adherence junctions or tight junctions? So first of all, we found that indeed we have a gut vascular unit. So these are the blood vessels here in red. And these are GFAP positive, so these are the enteric glial cells. You can see that they are sheeting completely the blood vessels here. And we also find pericytes that are stained with desmin and alpha SMA. And so we have a gut vascular unit similar to the brain. And the endothelial cells are sealed to each other by the presence of adherence junctions. So here you can see VE cadherin and beta-catenin. You can see here that they are present in endothelial cells. But we also find tight junction proteins like, for instance, occludin and ZO1, and we'd also see JAMA. But we also see some claudins like claudin 12, but there are differences with the brain, because in the brain, one claudin that is very characteristic is claudin 5. We don't find it in the gut. But probably this is because the gut vascular barrier and the blood-brain barrier are different, because in the brain, you really want to control what is entering. In the gut, you want the antigens to enter, the food antigens to enter into blood circulation, both for inducing systemic tolerance, but also for nutrient uptake. So then, yes, I said that morphologically it looks like a barrier. It does functionally it looks like a barrier. So we know that since in analogy with the brain, that in the brain you have beta-catenin, which is off when the barrier is not yet formed. And this is correlating with the increase of a protein which is called PLVAP, which is a protein present in the fenestre. But when the barrier is established, beta-catenin goes on and PLVAP goes off. So we know that when PLVAP is off, the barrier is established and beta-catenin is on. So we looked also in the gut, and we found this is a staining for PLVAP. The PLVAP is not expressed both in the duodenum, jejunum, and ileum, but also in the colon. But still we didn't know whether it's not expressed or it's not expressed because of the presence of a barrier. So what we decided to do was to challenge these mice with Salmonella. So we know that Salmonella is a bug in the mouse, Salmonella typhimurium, capable of disseminating systemically, so it gets easily into the liver and then eventually in the spleen. So what we decided to do was to challenge the mice with Salmonella, and at different time points, 2 hours, 6 hours, and 24 hours later, we collected the organs and we plated them for the presence of CFUs, but also we did the immunofluorescence on intestinal cryosections in order to analyze whether the PLVAP was expressed. So you can see this is the duodenum, 2 hours, 6 hours, you can start seeing the expression of PLVAP. But what is striking is in the jejunum that at 6 hours you see expression of PLVAP. I remind you that PLVAP in the brain, when it is expressed, the barrier is disrupted. And at 10-24 hours, the situation goes back into normality. This strain of Salmonella is not capable of surviving within the mouse. And the same is true in the ileum. So you can see that there is an increase of PLVAP, which is lasting a longer time. And this correlates with an increase of blood transaminases, which is a measure of liver damage. And it also correlates with the capacity of Salmonella to get into systemically. So as expected, we do see Salmonella in early time points in the Peyer's patches and in the mesenteric lymph nodes, but at later time points, we do see it also in the liver and in the spleen eventually. And this correlates with an increase of transaminases. So the gut vascular barrier has some similarities, but also some differences with the blood-brain barrier. Salmonella typhimurium can induce the upregulation of PLVAP, and this correlates with the capacity of Salmonella to enter the barrier and to disseminate systemically. So Salmonella is capable of modifying the barrier to its own purpose. One other possibility was the following, is that because Salmonella, we know that it's capable of crossing the epithelium, it's just the capacity of Salmonella to cross the epithelium and to face the endothelium that allows the Salmonella to enter the endothelium. So in order to understand this, we wanted to really see whether we had an endothelial barrier and whether this endothelial barrier was disrupted after Salmonella infection. So to do so, we went into in vivo imaging using 2-photon microscopy. So with 2-photon microscopy, we can go very deep into the tissue, and so we can analyze a whole intestinal villi. So if we put in the tail vein of the mice a dye that stains the vessels, we can see whether the dye is going out of the vessels. So the vessels become fluorescent, and if the dye is going out, then you can find the dye also in the lamina propria of the intestinal villi. And we can analyze this through two-photon microscopy. So the images you will see later on, you have the epithelium, which is not stained, but you can see nicely the vessels. So what happens if we first feed the animals with Salmonella? So we induce a breakage of the barrier, and then we expose the gut mucosa, and then we put the dye intravenously, and we see the effect of the extravasation of the dye by two-photon microscopy. So here is the mice that have not been treated with Salmonella. We used two different dyes, 4 kilodalton dye and 70 kilodalton dye. We could see whether there was a size exclusion of extravasation and whether Salmonella was, these are the mice that have been treated previously with Salmonella, if Salmonella is modifying the barrier. So you can see here that the 4 kilodalton dye can go easily, can extravasate easily. So 4 kilodalton molecules can extravasate through the endothelium. And if you first pre-treat the animals with Salmonella, there is not much difference, so it still can go through. But now you can see it accumulating in the intestinal villi, so this, in the lumen of the intestines. So these are the villi. You can see it accumulating. The reason being that when you have Salmonella infection, also the epithelium is broken, and so the dye can accumulate in the intestinal lumen. So what happens if you use the 70 kilodalton dye? Now, with the 70 kilodalton dye, you can see that this is completely retained within the vessels. It's not going out in the lamina propria after, under steady-state conditions. So what happens after Salmonella? After Salmonella, you can see now that the dye is going out. So these experiments are telling us, first, that we have a barrier and that the size exclusion is between 4 kilodalton and 70 kilodalton, and that after Salmonella infection, the barrier is disrupted, and you can see that the dye is extravasating out. So Salmonella is capable of not only crossing the epithelium, but also on disrupting the barrier. So what is the mechanism that is employed by the Salmonella in order to break the barrier? So we know that in the brain that the barrier is established through activation of beta-catenin, and when beta-catenin is on, some downstream genes are activated, like for instance axin 2. So what happens when you treat endothelial cells, primary endothelial cells, with Salmonella? So normally you already have some expression of axin 2, and after Salmonella infection, this expression goes down. And even if you treat these endothelial cells with Wnt3, so you activate the Wnt beta-catenin signaling pathway, this is reduced after Salmonella infection. So Salmonella can block the activation of beta-catenin. Because we know that Salmonella has several pathogenicity islands, so the SPY1 that allows the Salmonella to cross the epithelial cells, but also the endothelial cells, and then SPY2, which allows the Salmonella to survive within the vacuoles. We looked at the different SPY1 or SPY2 mutants, and we found that it is the SPY2 mutants, so this mutant here, that loses the capacity to control beta-catenin activation. So we used the SPY2 mutant now in vivo, and we found that the SPY2 mutant is not capable anymore to disseminate into the liver and the spleen, and it's not capable of activating PLVAP. You can see here, this is the wild-type Salmonella, and this is the SPY2 mutant. You can see that it's not capable of upregulating PLVAP. The barrier is present also in the human gut, so what we decided to do was to take human specimens. We separated the mucosa from the submucosa, and again you can see that these vessels are completely surrounded by enteric glial cells, and you also have pericytes. And what happens if you now treat with Salmonella? We use this model system in which we glue a cave cylinder on the top of the tissue, and this allows us to create a compartment where we can put the Salmonella, and the tissue is put on a grid and is taken into culture. And you can see that the tissue, if you look at PLVAP, I remind you, PLVAP down, there is a barrier. After Salmonella treatment, what happens that PLVAP is upregulated. So also in the human gut, we have a similar structure, and this is modified by Salmonella. So in conclusions, what we described is that under steady state, we have the epithelium, which is still, I don't want to make a wrong impression on you, that the epithelium is the most important barrier. So the epithelial barrier blocks the microbiota together with the mucus from entering the host. But even if the microbiota is capable of crossing the epithelium, it has to face a second barrier, which is the endothelial barrier. Not all of the bacteria are capable of crossing the endothelial barrier. So if we take an E. coli, we make it capable of crossing the epithelium, still is not capable of crossing the endothelium because it's lacking the SPY2, the pathogenicity element. And so once it's inside the host, and if the bugs are capable of crossing the endothelial barrier, they can disseminate systemically and get into the liver and can induce liver damage. So in conclusions, the microbiota diversity is dependent on IgAs. IgA levels are genetically determined, but also can be expanded by the microbiota. There is a gut vascular barrier that controls the dissemination of bacteria to systemic sites, and this is present also in the human system and is disrupted by the Salmonella. And these are the people that collaborated to the study, in particular Ilaria Spadoni, who worked on the GVB, and Elena Zagato that worked on the microbiota and the IgAs. And I thank you all for the attention.